Hello, everyone. Welcome to our iNaturalist MasterClass. We'll get started here in just a minute. We'll let people log on here for the next 30 seconds or so. You can see us, but we cannot see you. And so we'd love to hear more about you via words. So feel free to please type in the chat. Let us know where you're logging in from. We'd love to know where you're calling in from as people are logging in to the webinar. And then otherwise, we'll get rolling here in the next 15 seconds or so with our masterclass. As you're logging on, tell us where you're from. We'd love to hear or see your words typed in the chat if you are calling in from anywhere that's not where I am. Hey, Gwen, Salt Lake City. Awesome. Thanks for joining. Sandy, Utah. Fabulous. Great news. Well, as people log on, tell us where you're coming from in the chat. We'd love to hear you. Hey, Matt, Heber City. Nice to have you here. But I'll get us kicked off. Well, hello, Cody and Autumn. Hi, guys. Thanks for tuning in. Happy to have you. All the way from West Jordan. Hey, Christina, thanks for joining in. Okay, let's get rolling with our masterclass, everybody. Hello and welcome to our iNaturalist Masterclass, How to Make a Science-Worthy Observation, presented by the Natural History Museum of Utah. I'm Ellen Eiriksson, Manager of Citizen Science Programs at NHMU, and I'm joining you here from the Natural History Museum of Utah's Rio Tinto Center in Salt Lake City, Utah. In this era of virtual gatherings, it's important to reflect on our own physical sense of place, the land that we occupy, and to honor the Indigenous peoples who have called it home, acknowledging that all Indigenous nations are living communities. The Natural History Museum is part of the University of Utah, who acknowledges the Salt Lake Valley has always been a gathering place for Indigenous peoples. We acknowledge that this land, which is named for the Ute tribe, is the traditional and ancestral homeland of the Shoshone, Paiute, Goshute, and Ute tribes. The University of Utah recognizes and respects the enduring relationship that exists between many Indigenous peoples and their traditional homelands, and we respect the sovereign relationship between tribes, states, and the federal government. And we affirm the University of Utah's commitment to a partnership with Native nations and urban Indian communities through research, education, and community outreach activities. The Natural History Museum of Utah is committed to illuminating the natural world and the place of humans within it. And our citizen science programs connect people with the scientific practice, harnessing the power of community to learn more about the world around us. Tonight, we'll be exploring the wonderful and collaborative world of iNaturalist in celebration of the City Nature Challenge, a 4-day global BioBlitz that kicks off this Friday, April 29th. The City Nature Challenge is all about finding and photographing as many wild living things as you can. And we look forward to seeing all of the exciting things that you find this weekend. Over 400 cities around the globe are taking part. It's one of the largest BioBlitz events in the world. Last year, over 1 million observations were made on iNaturalist during the City Nature Challenge. We'll all be out this year too, making observations for our own city here in Utah. It's really a conglomerate of 8 counties that we call Utah's Wasatch. If you find yourself in Box Elder, Cache, Davis, Salt Lake, Summit, Utah, Wasatch, or Weber counties in Utah this weekend, make some iNaturalist observations. Any of them will count towards the City Nature Challenge, and it will help showcase Utah's unique biodiversity. I'll be out there, and I'm excited to see what you find, because I bet you'll be out there too. Today, I'm joined by 3 iNaturalist users who will be out during the City Nature Challenge also, and they're here as experts in some specific taxonomic areas. They'll be passing along some tips and tricks to all of you to help elevate your iNaturalist game. I'd love to introduce them quickly here. First, we have NHMU citizen science volunteer, who also happens to moonlight as a certified iNaturalist curator, and amateur arachnologist, Rebecca Ray. And Utah Valley University adjunct professor of biology and the resident expert on the evolutionary implications of plant and herbivores on ecosystems, Michael C. Rotter. And joining via the powers of modern technology, NHMU Vertebrate Collections Manager Katrina Derieg will be with us later in the hour as well. So the plan here is for the next hour to chat about some fun iNaturalist things. We'll all share some knowledge with you, and then we'll open it up for questions at the end, wrapping up by 7. So everyone can go home and have some, or you're probably home already, so you can enjoy some dinner. If you aren't doing it with us already, which is fine too, a little dinner and a show. So in case you're new to iNaturalist, let me give you a quick crash course. And to do that, I'd love to share my screen. Although I have about 500 windows open on my computer. Okay, so iNaturalist. In case you're new, here's the crash course. It's an online platform and application. Yeah.
Those black boxes.
Oh, thank you, Rebecca. Let me lose my Q&A here. There, is that better?
Yes, thank you.
Thank you very much. So iNaturalist is designed to allow anybody, experts and amateurs alike, to upload nature observations, typically photos or sound recordings that can all be scientific data. So what is an iNaturalist observation exactly? Well, it's a record of a specific wild organism or evidence of a specific organism in a specific location at a specific time. Like a weed in the sidewalk, an American robin on the side of a trail, a bee on a flower, or a deer track in the mud, or in this case, a quillwort, a snail, or a spider. Observations are designed to capture a photo or multiple photos of a single organism, and then geotagging it as a point on a map. Effectively, every observation on iNaturalist, and to date there are over 95 million of them, can be described as a point on a map. They are publicly accessible, searchable, and are a growing global database of biodiversity. So iNaturalist is a learning tool as well. Not only does it hold info on biodiversity, but it can help you learn about what you found. It has artificial intelligence. Oh, here we have the map of all of the people around the world. Artificial intelligence that can help identify what you've taken a photo of. So you're learning in real time what you've just found. Many times observations can be identified to the species level, but not always. Sometimes an observation can be labeled in a more broad taxonomic term, like a family or a kingdom. Either way, it's all still very useful data. So who uses iNaturalist? People like me, people like Michael, people like Rebecca. Lots of people can use iNaturalist. Many are amateur naturalists photographing the nature that they see in their yards or on hikes or on vacations. But many others are scientists or researchers, also like Michael, who are interested in nature and have some very specific knowledge about some taxonomic areas. They might be taking photos as well, but they're also looking at the images that other people have taken, and they help identify and verify the organism that was photographed. So iNaturalist is really like one big biodiversity online conversation, social media for nature nerds, if you will. You are exploring nature, sharing the things that you find, and then the platform is able to help you identify your observation so you can learn more about what you're seeing. Then the human iNaturalist community helps to further identify and verify your observation, making it a scientifically valid record of an organism, an important biodiversity data point. So when you take a picture of a bird at a feeder and you don't know what it is, iNaturalist will help you identify it using the information that you have uploaded, your photo, the location, the date and time. And other iNaturalist users will help verify or change this ID based on that same information shared in your observation. Then your bird picture becomes useful and accessible data, which I think is a pretty exciting thing. But are all observations created equally? If you spent any time on iNaturalist, especially time making IDs or verifications, you've likely noted that while all observations can capture the same information, like a photo, date, time, location, not all observations are able to communicate the same amount of information. So some observations are in fact better than others. They tell a richer story, they paint a clearer picture, they pass along more information. Today, in our iNaturalist Masterclass, we're going to touch on some key points that can elevate your own iNaturalist observations to being more ideal, scientifically useful observations, the best that they can be, in a way. Disclaimer, an iNaturalist observation certainly doesn't have to be perfect, and many times they can't be. As iNaturalist users ourselves, we know very well that many factors can influence an observation. A bird flies away, the insect moves too quickly, the large mammal in the distance is so far away that you can only see a silhouette. Maybe the sun is shining too brightly or not brightly enough. The wind is blowing. Your flower happens to be on a cliff and it's really hard to get to. Knowing that, we just know that being safe yourself and respecting wildlife is the most important thing when it comes to making an iNaturalist observation. So that said, get the best shots that you can, the best photos that you can, and know that any observation that you make is still contributing to global conversations about biodiversity. So tonight, we're here to get you thinking a little differently about how your picture or multiple pictures, in the case of an iNaturalist observation, can tell the best story possible to empower you to pass along the most information that you can in an observation. We'll focus on 3-ish taxonomic areas: arachnids, plants, and vertebrates, but we'll dabble in some other things. Know that many of the tips that you hear tonight will apply to a wide variety of organisms. To start us off, I'm going to pass it on to Rebecca. Rebecca Ray is going to start everything off. She's our resident arachnologist. Rebecca, I'll leave it to you.
Thank you very much. All right, I'm going to share my screen here. There we go. All right, so I'm going to talk mostly about taking pictures of spiders, but I want you to know that a lot of what I talk about will translate to insects as well. There are lots of spiders and insects, and although it's a tiny world, it is a vast world. So speaking of that, did you know that there are over 600 species of spiders in Utah? That's a lot of spiders. So of course, a lot of those are going to be hard to identify. Some of them are impossible to identify to species from photographs alone. They actually require looking under a microscope to get them to species identification. But if we make really good observations, some of them we're able to identify them to species, and we can at least for sure narrow down the identification and get closer to species level if we have good information in the photographs and what we include. So when it comes to naturalist photography, like Ellen was saying, you don't have much control of your subjects, right? A lot can happen, but here are some guidelines that you can follow. So if you can only get one type of shot, I would try to get a dorsal view. So when I say dorsal, I mean the top view, looking at the top side. So think of the dorsal fin of a dolphin or a whale. That's the top. This is looking down on the spider or the insect. You're going to see the top of it. And I mean looking at the whole organism. So with a spider, that's going to give us the overall shape of it. We're going to see the length of the legs and the proportion of the legs, and we're going to see any markings or patterns on the top where they're most likely to be and where they're most likely important. So this is the most valuable shot that you can take if you only get the chance to take one. Now, like I said, you don't always have control. Sometimes you can only get a view of the underside or a view of the side, but if you can get that top shot, that's going to be your most valuable shot. Now I'm going to give one sort of precursor to all of this, and that's to try to get close. So we often see shots that are from really far away because we know lots of people are afraid of spiders and they don't want to get close. I'm going to challenge you all to face your fears, because actually I would say that your biggest danger from the organisms themselves is scaring them away. So of course you have to be careful, but what you need to be careful of more is other things around. So you want to watch where you put your hands. You want to watch for things hiding under rocks like snakes, things like that. But these little organisms themselves are really not going to come after you. They're not going to do anything to you. They're more likely to run away. So you want to be really calm, really quiet, and move slowly. You want to get as close as you can to get the good shots. So we're going to start with that dorsal shot. Now, if you can get two types of shots, I recommend trying to get key details like the eye arrangement and a closer, better look at the markings and patterns on the abdomen and head. So when I talk about the eye arrangement, spiders have their eyes, usually 8 eyes. Some of them have 6 eyes or even less. There are actually spiders with no eyes, believe it or not. But the shape of how those eyes are arranged can actually give us immediate information about how to identify them. So this shot right here is of a clubionid, and this is a specific type of spider that will wrap a leaf over its nest or egg sac. And there are other types of spiders that do that as well, but they don't have the same eye arrangement. So I can tell from this look of the spider exactly what type of spider this is. A wolf spider has a very particular eye arrangement that is not like any other spider. So when we can see a good view of the eye arrangement of a wolf spider or of a jumping spider, we can know immediately what kind of spider that is just from its eyes. So if you can get a good look at those eyes, it will really help identifiers. Now, you may not need to get extra shots of these details if your first shots of that dorsal view was really in good focus and was high enough resolution that you can zoom in and see those details. But sometimes your focus may not have picked up all of the details in one. It's hard sometimes to get the focus on each of those spots in one shot. So you might need to take a few shots focusing on more than one. We all know that insects and spiders don't like to hold still. So sometimes you have to take multiple shots trying to focus in different points. Now, if you're able to get more types of shots, and get 3 types of shots, this is where I say try to get more angles. So I would say try to get the pedipalps. Now, spiders have 8 legs. We know that they're the 8-legged, we call them 8-legged freaks, right? So our insects have 6 legs, arachnids have 8 legs. But the pedipalps are those extra ones that sometimes look like a 10th pair of legs in front of their face. And you can have the jaws and then another sort of looks like a pair of fingers. They use them kind of like fingers sometimes. Those pedipalps are where actually the males, a mature male has their genitalia at the end of their pedipalps. And so that can be really, really useful information for not only knowing the gender of the spider, but that can help to identify what the species is of the spider. So we can get important information from those pedipalps. If you can get a view of the spinnerets. So the pictures I have here are of a ground spider, and they have sort of barrel-shaped spinnerets that come off the end. They look kind of like tailpipes. And that's a very particular feature of ground spiders. But if we don't get to see those spinnerets, then we might not know that it's a ground spider. If you can ever see the ventral view, so we have dorsal on top, ventral is the underside. So it's not always useful, but it can be useful in some spiders, especially the orb weavers. So these are the ones that make the big, flat, round, geometric shaped, the beautiful webs like Charlotte's web. Those spiders, and they're often easier to get the underside, the ventral view, they have very useful patterns on their underside for identifying. And you never know. So wolf spiders sometimes have really useful markings and coloration, color patterns on their ventral, their underside, on their venter. So sometimes it can be really useful to get that view. Now, not all spiders are easy to get a view of their underside. They're not going to like being flipped over. So for instance, in these pictures, I've gotten the spider into a petri dish with a top on it. I'm able to just lift it up and take pictures of the underside. Now they still like to run around in there, so it might take several shots to get it to hold still. Some people will carefully capture a spider in a plastic bag and just really gently get it to where you can get it held still in the plastic bag, and then you can flip it over and see its underside. And then you can still release that and it's unharmed. You can also get a view of its side, the side view. So that will show us the different angles of its different parts. So you can see how tall different parts of the head are, the cephalothorax, and different shapes of it can mean different things. And most fun is the spider's face. And that can really come into play in things like the jumping spider. So they have different colors and different features, and even the mature males of jumping spiders can have different features they use in mating that make them really fun to look at and attractive, and really make them stand apart from the different species. So if you get to spend more time with the spider and get to take those views from different angles, that can really help in identifying them. And you get to learn a lot more about them the more you get to see them from different angles. So even if you don't get to see them from lots of different angles, don't forget to snap a shot of if they have a spider web or an egg sac or sacs if they're nearby. So be sure to look around if there was a spider and see if there's anything that belongs to them, because that can also be really useful in identifying. Now, they might just be on the move and didn't have anything that belonged to them, but if they do have something that helps identify, if they're near their home or have a trap laid or something, spider identifiers can use any of that information. Be really useful. Now, in identifying arachnids and insects, and really all arthropods, it's all about capturing the information. So I may not know about another type of insect. I've studied a lot of insects, but for instance, I did this observation of a cicada that I had found. I was in an area where these cicadas that were just beautiful. They looked almost like they had been painted, and there were dead ones all over the ground. So I had the opportunity to pick this up and take pictures of it from any angle that I wanted. Well, not being familiar with it, I didn't know what would be useful for an identifier. So I'm just going to show you how, let's see, how I chose to photograph this one. So I took this dorsal view, took a side view, got kind of the side and underside. This is more of the underside of the face. So it was really just trying to pick up. I was sure to get the wings because wings have different venation patterns, the veins running through them. I was really trying to show any details that I could think of just because I didn't know what an identifier would need. And that's really the key. And as it turned out, here came someone that knew exactly what the species was. And I was able to look it up and look into any of the options in the area and confirm that that was it. I was really excited to get a species level ID on that one. That was a neat one. So a lot of times people will just take one photo and say, cool, I got my one photo of that, my observation is done. And it might be a beautiful photo, but it might not have the information. And you might find that that's the one note that comes back most often from identifiers is, do you have any more photos? Because they're just not able to identify it from the information that's there. So finally, my challenge to you is to get closer to your tiny subjects. In order to see the information we need, we really want to see them as close as possible. So try not to be afraid, try to get as close as you can to see what you need to see, and don't be afraid. But as always, stay safe. So thanks and have fun.
I love it, Rebecca. And under your tutelage, I truly, I can say that it works. So be brave, get out there, people, and get close to some spiders. And they're a lot more friendly looking when you get up close than you'd think. Really. And I now know where to find them out in my house. And it's fun for me. Janine was asking while you were chatting about it being okay to temporarily capture a spider in a specimen jar to get multiple shots. Is that something that works pretty well and it's okay for the spiders to do?
Yes, absolutely. As long as you are being careful with the specimen, respecting it, and then putting it back where you found it.
And then James was just asking, and I think this ties to what you were talking about, so maybe we'll just touch base on it. He was asking, do you have any recommendations for agreeing with an identification when in reality you don't really have an idea? Are you supposed to are you supposed to do that or not supposed to agree?
So I agree when I have done the research to follow it up. So if I go and look into what the person has suggested as the identification, and I mean, I will really look into it and look at any other possibilities in my area, what other genuses, genera are here, what other species could be here. And if I feel confident that I've really looked into it and I know all other possibilities and it's not just a guess, then I feel like it's okay to agree. But if I really don't have that knowledge and it would just be a guess on my part, then no, it's really not okay to agree.
It's a great insights into some iNaturalist best practices. I think that's awesome. Thank you, Rebecca. If other spider questions or any kind of insect questions come up for the rest of the talk, feel free, everybody, to put it in the chat or the Q&A functionality at the bottom right corner of your screen. Thank you, Rebecca. We'll touch base with her again later, but let's turn it over to Michael. I'll let you chat a little bit about some plants.
Cool. Thanks, everybody. And this is one of my favorite parts of the year, doing a City Nature Challenge, because a lot of things, there you go. Sorry, I'm just kind of fumbling with screens on my end real fast. A lot of things that the City Nature Challenge does really helps a lot with my research. I'm relatively new to Utah. About the last 2 years, I came here to take this position at Utah Valley University. And iNaturalist was really my first go-to of it. I really depend on that to find populations of plants I study, as well as just kind of for fun, I like to ID plants and insects in particular. And so iNaturalist is probably, I think, the best social media out there, and also the least toxic for sure of all of them. So, you know, I always like to just kind of pull up my profile and say, you know, come follow me on iNaturalist. I always love to interact with local folks and what you're doing out there. And, you know, a lot of times if you find something cool, you can bet I'll go check it out too. A nice thing about iNaturalist too, if there's an organism you're interested in seeing in the wild, you know, you can kind of put out a list for it and try to go and find it if somebody reports it. You know, I have on my favorite list here, I have things like beaver that are listed because I have a student who's very interested in studying beaver forage preferences. And then I have things that I'm interested in studying invasive species. So I'm studying this watercress here, as well as things like nightshades, which I'm kind of moving into with my research. And then for fun, I always follow monkey flower because that's what I did for my dissertation. But iNaturalist is just this great thing to use, especially as a researcher. And as a researcher, I do depend on great observations by the community out there, especially the amateur community, because, you know, the dirty secret is the amateur botanists usually know a lot more than the professional botanists out there. And so it's always great to be able to collaborate like that. So when you're making good plant observations, I kind of have 3 rules I like to stick to of it. And if I can change my PowerPoint, and those are kind of look around, keep it sexy, and eat your greens is kind of how I like to mention them. The first one is look around. It's always good to get a picture of your plant in good general context of what it's growing in. So today I was kind of out in my break, just kind of getting out of my office. And I always like to take little iNaturalist photos of just the weeds growing here on campus. Our grounds crew is a little too good, so it's hard to find the weeds all the time. But I found this one in kind of an abandoned little lot. And the first thing I'd like to do is just take a big picture of it. That way we can see the habit it's growing in, what it looks like in general, and also the habitat it's in. And so by this picture over here, you can kind of see it's just kind of a cruddy little hard pack area. And that's important for a lot of our species ID to get a context of that. And you can also tell it's, you know, a kind of a shooting up plant, has leaves on the bottom, and it's going well on it. So understanding that, looking around what it's like is key on these plants. The next part, I like to call it keep it sexy. You know, it's Utah, so we can't get too sexy with it, but these plants have a lot of parts that are really important to ID, and a lot of them are their sex bits, their flowers or their seeds, the kind of consequences of that sexual interaction in plants are going to be key for IDing them. And so what I like to do on those pictures is I like to get right up close as best as I can with my camera, usually just on my phone, and try to get a good snapshot of it. There's a lot of little things you could buy to like kind of macro lenses to that strap onto your phone. Sometimes I always have a little small magnifying lens that I keep on me. You can even take pictures right through the phone. That works pretty well on it. And so those are always good to get as detailed as you can on that, because a lot of times we'll only be able to ID that plant based on that flower. The other thing I like to do when looking at little parts is throw a little ruler in it if you have one. If you don't have a ruler, anything like a pocket knife, a coin works really well. And that will give us kind of a context of what size those bits are. And then the last bit I like to say is eat your greens. Look at the green part or the leaves of those plants, because a lot of times, even if the flower might be a little obscured, the leaves can tell us a lot of really good information about that plant. And there's also some kind of new research coming up that we're actually interested in the size of those leaves and how they change throughout the years and where you're at too. So getting those leaves is a really nice little treasure trove that can really kind of push us over the edge to get a good ID on it. So they'll keep those 3 rules in mind when you're kind of taking pictures of plants. The last other little bit I will say about taking plants to keep in mind is that, you know, a lot of times we'll run into cultivated plants, we'll run into rare plants, and also there might be just kind of random plants that we don't really know what to bucket to put these in. And my philosophy on iNaturalist is everything is useful. Even the cultivated plants are great to have in there. In my own work, one of the things I'm interested in is how do cultivated plants escape and start becoming invasive. And if we have good records of cultivated plants, we might know when they were first introduced and when they kind of started to spread. And so those are really nice things to keep track of. Today I was out again and decided to take a picture of redbud that's on campus here. And we have a ton of them blooming. They're beautiful and they're all cultivated. And so I took some observations of that. And then down here, you're going to have this button. Is it captive or cultivated? And just click it. And that will allow us to easily sort on the back end, you know, what are we dealing with there? I also like to put in notes occasionally on there. So in here, I had about 20 plants in the area. So we kind of get an idea of that population size too. So you don't have to take pictures of every single redbud, georeference them. You could just say in general, in this area, that's about what we have in there. The other thing on kind of on the flip side of this cultivated plants is sometimes you might find really rare stuff out there. In fact, iNaturalist has a great history of finding species that we thought were no longer in the area and they just show up. And so if you happen to do find a rare plant, you might be a little concerned that there could be some poaching issues because we definitely have a problem with that, especially here in Utah. Any orchids or cactuses that are rare that I take photos of, I always get a little concerned about that because we do have poachers that can use these platforms for kind of ill-gotten purposes. And so what I like to do a lot of times is I will click on this location visibility here. So just this weekend, I found this rare plant down near Capitol Reef National Park, and I took a picture of it. I wanted to share it. And but I knew that people would potentially poach it if I shared it. And so I kind of obscured the locality of it. And so only certain people on the back end with those privileges, like land managers for that area, would be able to see that location. So it's still helpful data, but you don't have to worry about kind of the negative consequences of that. I also like to point out that this is an awful picture of this plant. It's blurry. I didn't take multiple pictures of it. There's no scale in it. This thing's about the size of my thumb. But you know, what I like to say is that, you know, even the professionals out here, we're not taking great pictures all the time. In fact, in this, I was kind of rushing to take the picture and catch back up with the students I was with that weekend. And so it turned out blurry. I didn't even realize it until I got back home. Unfortunate, but it's still really useful information and something that people like the Park Service of the BLM would love to know so they can keep an eye on these rare species out there. The last little bit too with kind of some of my plant tips is sometimes when you're taking pictures of plants, you're going to find other stuff on there. A lot of times these plants have pollinators coming to them, they have things eating them, and those things are great. And I think it's best to just have both. And so this is a great time that you can take advantage of your well-gotten first shot and kind of double up your observations with. So during my dissertation work, I was looking at plants that are insects that eat monkey flower here. And so a lot of times I would have pictures of the monkey flower and I would post that as a one observation and then just use that same picture to post again on another observation that I had this beetle kind of that was there chewing those leaves or that flowers that day. And so those observations are incredibly helpful for people like me that are really interested in these interactions because we can take those geolocations, those dates, and kind of match them up together and get really good records of when these insects were eating these plants or when a pollinator was coming to visit. And so it's really just excellent practice to do if you do get lucky to kind of have those fun interactions there. And then also in the notes section, you can just reference kind of each other's observations on that. The last little bit I will say is that we do have some plants that you want to be careful with when you're handling them quite a bit, and these are always good things to be aware of. The first one that I'm sure a lot of us have an unfortunate interactions with in the past is poison ivy. Around this time of year, especially in our foothills here in Utah, it's not going to look that prominent. In fact, you'll usually see these kind of twigs, but usually they'll have these kind of white berries on it. And they're not that active this time of year. They're going to, in a couple weeks, you'll really want to stay away from them. But it's always good just to not get involved in it if you can, and take those pictures from a distance. If you don't put a ruler in that picture, no one's going to blame you on that one. So document them, but stay clear. And then later on in the year, they get these very prominent leaves. The leaflets 3, let it be, and there'll be a little glossy red to them too. So just stay away from it. We also have a couple of species locally that are also things to keep aware of, but ones that we're really interested in documenting because they are becoming a spreading problem. Donkey's tail spurge is in the news kind of once a year, unfortunately, and usually it's because a dog or a small child ate some and got really sick from it. But these are kind of big, thick, succulent plants. They used to be planted quite widely as a garden plant, and now they're escaping into our foothills. Where they can cause very serious contact injuries if you break them, and they have very milky sap that can cause rashes on your hands. In fact, last summer, I grabbed a whole bunch and got a bunch of the sap on my hands and had some really nice blisters for a couple months. So they're really no joke, and you do want to stay away from them. But getting good documentation of them is always great. The best thing are they're very unique to our area. So, getting a good kind of habit picture like this is going to be sufficient. The other little bit kind of to watch out for are parsnips. This is another non-native that seems to be kind of spreading into our area. And a lot of members of the carrot family, which have these big kind of umbel-like flowers to them, can cause also severe contact dermatitis too, especially the sap. Again, if it gets on your hands, the sunlight hits your hands, it can cause a very big rash on your hands or your skin. So you want to be a little careful with these. Again, and any of the kind of carrot family stuff, just be a little cautious of. But they're not going to necessarily kill you or anything like that. But they can be a very unpleasant experience. And last little bit, I want to leave you guys with some of the stuff that I think is coming up right now that is kind of good to note. We have a lot of our blue mustards, which is this non-native mustard plant right here. Mustards are really nice to ID because they have these very prominent 4 petals on them. So you'll be seeing a lot of those, especially in our cities and things like that. Our red stem storksbill, which they have these 5 kind of geranium-like 5-petaled geranium flowers on them, are usually growing in our sidewalks right now. In fact, a lot of my students had questions on them today when I was out with them this morning. And then also one of my personal favorites, grape hyacinths, a lawn weed, but one that's kind of found its home here, is really popping up quite a bit. And they have these kind of very unique flowers to them. And then a few other ones to kind of point out too is our trees are really flowering too right now. They have very obscure flowers, but they are kind of pretty in their own rights if you get up into them, especially our maples, especially box elder, which you'll see around towns quite a bit, are flowering right now. Some of our bed straws, if you grab one of these plants, they'll feel a little sticky because they have little tiny hairs. And I always like to throw it on a student's back and then have them carry it around until it's time for me to pull it off their back and talk to them about bed straws in the field. And then we also have just tons of other mustards coming up right now. So if you're into mustards and want to ID them, get out right now. Look for all these small little white and yellow flowers out there, and they're always a lot of fun to ID. And you can even ID them just a mustard, and you can bet if you're in Utah, I'll probably be jumping on iNaturalist and helping you ID them. So basically, plants are great. Get out there, start looking at them. It's really just the most exciting time of year to be a botanist.
Thanks, Michael. Dr. Michael Rotter, everyone, Utah Valley University professor and wonderful human.
Oh, thanks.
Gwen, I see your question here about the app and the web browser version of iNaturalist. So maybe we'll touch on that at the end. That they essentially are the same. They just look different, and you know they're the using it on your phone. Of course, like the camera's right there with you if you want to use it. I think is the main difference. But before we talk about maybe some of those things, I'd love to get into some vertebrate talks as well. We've been hearing some awesome things about arachnids, plants, and Katrina Derieg, who is the collections manager here of the Vertebrates collection at the museum, is out in the field right now doing some research and specimen collection and probably making some iNaturalist observations as well. And so she can't be here with us in person virtually. So she's recorded a few minutes of chatting about some cool things to keep an eye out for when you're observing vertebrate animals. And so I'm going to play that video for you now. And then when the video is done, we'll get into some questions with everybody. So here's hoping it works. It will, probably.
Learn how to make high quality observations on iNaturalist ahead of this year's City Nature Challenge. I'm excited to share some tips and tricks on how to make the best possible observation when observing vertebrates. I'm mostly going to focus on mammals and birds, but some of these tips will be relevant to most of the taxa that you'll be observing during the City Nature Challenge. Now, this presentation is not about how to identify different animals in particular, but it's more about how to make observations that show enough distinguishing features on an animal so that others can review your observations and then make accurate identifications. And the main thing I want you to take away from this is that you don't have to be a perfect naturalist. You don't have to identify every animal you see. And this is why iNaturalist is so cool. You don't have to know everything, and the community can help you identify the things that you see. Also, iNaturalist makes use of machine learning or artificial intelligence to guess what your photos are. So taking better photos will help the AI algorithm accurately guess what you've observed. And so in this webinar, we'll cover a few basic do's and don'ts of making an observation on iNaturalist. The most important thing that you want to do is take multiple pictures, and I'll show you why in a second. It's also important to include context, so that can mean habitat that shows us more about where you found the animal, and then including an object or ruler for scale could be helpful, especially when making an observation of signs like scat or tracks. And the main thing I see that makes an observation less useful is when people only take one photo. And so we don't get a lot of different features to look at, or people will zoom way in and the photo becomes too blurry. And I'm going to show you several examples. A lot of them are my own, good and bad examples. So here's a great example of why you should take more than one picture. This is the cliff chipmunk here. This is a common chipmunk on the rocky cliffs of the Southwest. And while this first photo here may look like a really nice photo, the second one shows the more distinguishing features of this species. So cliff chipmunks are mostly gray and have very faint stripes. That's difficult to see from this first photo. So does that mean that I should not include the first photo in my observation? No, I think it shows the ears and the forelimbs pretty well. And at the end of the day, more photos will always be better and give people on iNaturalist more to go off of. This is a photo of a mule deer that I took from my office at the museum. And as you can see, the photo here on the right is far too blurry to really know what we're looking at. The photo that is less zoomed in, though, it's more obvious that there's a deer in the photo. And so this is another good example of why you should take many, because if I had only taken this one super zoomed in photo, I would not really have a good photo to upload to iNaturalist. And now I want to touch on some specific groups and some key features to make sure to photograph. And so on the left here, we have 4 species of squirrels, and the 2 on the top are different chipmunk species. And on the bottom, we have the golden mantle ground squirrel and the Uinta ground squirrel. And so with chipmunks, the stripes are usually a distinguishing character, like with our cliff chipmunk here that I already talked about. But some have really faint stripes, some have dark stripes, some might have more red or more gray stripes. And then other ground squirrels, like the golden mantle ground squirrel, has stripes, a little bit different looking from the chipmunks. But the Uinta ground squirrel does not have stripes. And so So if I had just taken a photo of their faces, it might be difficult to tell which ground squirrel we have here. And on the top right here, we've got a deer mouse. On the bottom right is a wood rat, bushy-tailed wood rat. And I want to point out that these photos both clearly show the tails, the ears, and the eyes. The tail is super important for identification on a lot of rats and mice. As noted in the name here, the bushy-tailed wood rat, it has a very bushy tail. So that's helpful in figuring out the ID. Also, some mice and rats will have really big ears or maybe small ears, pointy ears. Same for eyes. Some species have really large eyes and some have smaller eyes. And these features can really help narrow down the IDs, even if it's just a genus. And so for some key features on birds, birds can be tricky because they can be flying really high up or perching high up in trees. One important feature is bill shape. This is helpful if colors or patterns are obscured in the photo. So this grosbeak on the right here clearly has a finch-shaped bill. Grosbeak is in the finch family. And so you can immediately tell that it's a type of finch. And then this curved bill thrasher here, you can see it's nice curved bill. So that helps narrow it down, even though the colors might be kind of unclear or the patterns on the bird. And oftentimes you'll see birds in flight. So the shape of the silhouette of a bird can be really helpful for distinguishing species. And again, this is when you want to take multiple photos of the bird, so you make sure you have a few good shots of different flight angles. And also, iNaturalist has a place for notes. So it can be really helpful to write about flight behavior. So was this bird soaring or diving into water, flapping its wings rapidly? All those things can be helpful to make a more accurate ID. And sometimes you might come across just a skull or bones. And in some cases, this will actually be the most helpful thing for making a correct identification. Mammal skulls in particular have a lot of distinct features on them. So it's important to capture them all. Again, taking multiple photos will be key. So photos of the front, top, back, bottom, showing teeth, that's really crucial. Other important features to show, particularly on mammal skulls, is the zygomatic arch, which is this structure here. That's basically the animal's cheekbone. Also, the back of the skull can be really helpful in determining species as well. And in this example, they actually have, a complete skeleton. And that's really helpful because you can see the animal's claws and see this is clearly a squirrel. And lastly, including something for scale is really helpful. So we can see how large the animal is. So this person, uses their hand, but something like a pen or your hat while you're hiking, something like that could be really helpful if you're, not carrying a ruler around with you everywhere you go. And in addition to animal remains, you might only come across animal signs. And these are great observations as well. I want you to feel empowered to take these kinds of observations because there are tons of them on iNaturalist and they are just as useful. And so you might see tracks, scat, or maybe nests. And really, I have the same advice as before. Include something for scale, like a foot or maybe your hiking pole. This person here apparently does carry a ruler around with them everywhere they go. But with tracks, the important thing is to capture the gait or walking pattern. So if you see multiple tracks, the important thing is getting a picture of those multiple tracks. You can see how this animal walks. So like rabbits and hares, they hop. So they have a pretty distinct gait in the snow here. Lizards have distinct tracks too because they have the tail drag. And then I also included here a wood rat nest and some fecal pellets. And this person used their hiking pole to show how large this nest is. And we may not be able to tell the exact species from this without knowing the location. Of course, we do know the location with iNaturalist. But this is still a great observation that shows evidence of an animal. It's definitely worthy of making an observation. And I also want to point out that including multiple signs makes for a really good observation as well. So this was a moose that I saw a few weeks ago. There were some really obvious tracks in the snow and some coarse, long brown hair left behind. And I was also lucky enough to see the animal itself off in the bushes. And so bottom line, more information will always help people to more accurately identify the species in your observations on iNaturalist. So go nuts, take lots of pictures, take pictures of everything. I want to thank you so much for listening. While I can't be there to answer questions live, my colleague Ellen is very knowledgeable, and I'm sure she'd be happy to answer any additional questions that you have. I hope you have a blast during this year's City Nature Challenge and beyond. And as always, thank you for being a citizen scientist and contributing to this amazing resource of biodiversity information.
Thank you, Katrina, wherever you are in the wilds of Utah. I love how she says, enjoy it, go nuts, just take some pictures. I think that's great. And you've basically heard Rebecca and Michael say the same thing during our time with you today. So if you can take anything away from this, go nuts, take a bunch of pictures, have fun, enjoy it. Using iNaturalist is a really, really fun thing to do when you're outside. And I suspect the more you use it, you'll realize that you start to look at the world in a slightly different kind of way. And it's a pretty fun thing. We've got a couple minutes together here still, and I'd love to get to questions that people have. So if there are other questions that have come up, feel free to throw them into the Q&A or the chat box. I'll revisit Gwen's question here about, she says, how does the phone app differ in how it's used? And I assume you mean from the online platform. You know, iNaturalist is the same on a phone versus using it on a web browser. There just are different functionalities. And so I'd say the app is a little more simplistic. It has the ability to take a photo right in the moment from the app if you'd like to. I personally use iNaturalist almost exclusively on my phone. I take photos with the camera app on my phone and then upload them into iNaturalist later. So I don't use the camera functionality on iNaturalist typically. But things like, you know, browsing maps and looking through lots of different observations to help identify are a little trickier on a phone. And so using it on a desktop is way more user-friendly in that way. Rebecca, I know, uses a digital SLR almost exclusively when she's taking photos of things. And so uploading it to the iNaturalist platform is essentially essential on a web browser platform instead of on a phone. So Rebecca, I know you primarily are using a web browser when you're doing iNaturalist stuff. And so if you're using a digital SLR, it's a lot easier to do batch uploads and that sort of thing too. But all that information goes into the same place. Any other questions from the group? Feel free to put them into the chat. I had a question this is for both of you, Rebecca and Michael, about tips on ways to sort of train yourself to look for certain things. Like, how do you find tiny spiders? How do you notice, like, how do you find a rare plant when you're on a hike? Any suggestions on ways to just sort of train naturalist eyes to being more observant in that way?
Well, I'd say that I have learned not only to look everywhere, and really your brain gets trained to look in a different way, but also you learn to look in different places. So, I mean, if you take the time to look at, I know you've had this experience, Ellen, when you look at, for instance, a group of flowers with normal eyes, I'll say, you see the flowers, and the longer you look at them, if you look closer, you may start to notice, you know, some buzzing insects. I used to think the tiny things were just flies and, you know, go away flies. I'd see the bigger things were bees. And as I learned more and noticed more, I learned that there were tiny bees, and I learned there were tiny wasps that weren't scary wasps. They were cute little wasps. And, you know, I learned that there were all these, I mean, the biodiversity around one flowering thing is amazing. And then I can look at a single flower sometimes for 20 minutes and still 20 minutes later find an insect I hadn't seen 20 minutes ago, because they're hidden in little crevices. And there's a rove beetle and a crab spider that is camouflaged waiting on a petal. And it just takes eyes that see, it takes looking at it a different way and waiting and, maybe taking a different perspective, but also turning things over. If you look underneath something, and I like going around my house, and along the edge of the house, you pull the grass away, and you'll find little creepy crawlies along the edge that maybe you didn't know were there. And turning over rocks, that's an old, kids like to do that, and you find things crawling under there. But just the more you look, the more you find.
Yeah, and I would very much echo that. I mean, the joke is, like, if you go, all my friends have with me, if they go on a hike, you're never going to get out of the parking lot, because you're always just, there's everything, there's so much around you. And if you slow down and just look at an area for long enough, you're going to find something that's hidden there. That tiny cactus I had, we were sitting at this spot for about a half hour before anybody saw it. And it was like right in front of us, but they're tiny. So, sometimes if you have just an extra 10 minutes to spare, just look at a patch in your lawn for a bit and just go through every blade of glass. There's a lot of little treasures hiding in there. And even the grass itself can be pretty interesting organism to observe. So just slow down and really take your time is my best advice on that.
I love it. And I think that really is like the iNaturalist way. Just, take your time, just look a little longer, stay a little longer, and you're going to find some pretty cool stuff. And that certainly has been the case for me over the years that I've grown to really, really love the platform. It's a fun thing. And so I'm so excited for people to get out this weekend and look for some nature with the City Nature Challenge. Gwen, you asked a great question if the museum's doing any activities or events with the City Nature Challenge. This is the first one of the week. We're kicking it off with our masterclass, and then we've got many things on the docket coming up. We've got a wildflower walk here at the Natural History Museum tomorrow evening, and then on Saturday, we'll be doing the City Nature Festival at the Tracy Aviary's Jordan River Nature Preserve. I'll be there talking about iNaturalist. We've got simultaneous awesome events happening that same day. So Michael will be doing an awesome bike trip along the Provo River to look at plants. All of this you can find all the info about all of this on our website, nhmu.utah.edu. Look for City Nature Challenge and you'll find lots of great stuff about all of our upcoming events. We're hosting an iNaturalist Nerd Mixer this Sunday here at the museum. Come up, hang out with me. I'll be poking around the building and we'll do that. Yeah, maybe we'll never leave the parking lot. We'll be out of the parking lot to start with, but maybe we won't get that far. And then we've got some fun things cooking next week to help ID a lot of the observations that are made during the City Nature Challenge. So lots of great ways for you to get involved personally with us. You have the opportunity to really engage with all of us in person this weekend during various events, and then of course virtually as well. There are lots of great opportunities to connect. Key takeaways from today for me: take multiple photos, put them when you're making an iNaturalist observation. Think like an identifier. What would somebody who wants to identify that species need to see, and try to get your brain to think about it in that kind of a way. iNaturalist makes a great break when you're at work. Take some time to get outside and take a couple pictures. Consider scale when you're taking photos. Maybe have some materials with you, like a ruler or some kind of object that's a known quantity. And then, of course, enjoy it. Have a really good time. Don't stress about making the perfect iNaturalist observation. You'll find the best ways for you to make an observation. Enjoy it. And remember that the more information that you share, the better. We'll all be out there with you at the same time, enjoying all of the things that you're exploring. We are so excited to see everything that you find. Oh, and details on the mixer on Sunday. I'll put a little thing in the chat right now. How about for you, Jennifer, for our nerd mixer? We'll be meeting at the Natural History Museum of Utah, 3 p.m. We'll be on the south side terraces along the Bonneville Shoreline Trail. I'll have signs out. All you have to do is navigate yourself to the Natural History Museum of Utah, park, walk up to the building, and then there'll be some signs that will point you in the right direction. And really, I'll be out there with my phone in hand and some cool materials you could borrow to catch some bugs. And of course, I can't find the link for it right now because I have too many windows open. nhmu.utah.edu, and you'll see our nerd mixer up on the website. Thanks to everybody for joining us. We've kept you a little bit longer than I meant to, but it's all for good reasons. Stay tuned for more fun stuff, and we're so excited to see what you find this weekend. Happy City Nature Challenge, everybody.